Thank you, Ian. Moving right along, Eric Nuermberger is going to share with us the preclinical side of things with the exciting new work coming out of the hollow fiber work and the animal models. So, Eric, take it away.
Thanks, Klaus. It's a privilege to be here and to be able to update you on the work of the preclinical and clinical sciences work group. And I'll just take this opportunity to thank Deborah and my other co-chair, Christine Sizemore, for all of their very helpful input over the years. And so what I'm mainly going to do is just try and give you an overview and update on the progress that's been made in this area, focusing mostly on the hollow fiber model qualification process and what's gone on in the post-qualification era, and then to tell you a little bit about what we're actively working on now, which is going through a similar evaluation for the sterilizing mouse model. It looks like these slides are a bit bleached out, but hopefully we can read these. So I'll thank Dan for going through much of the information here, describing this big gap that has been made apparent by recent phase 3 results between what we're doing in preclinical and early clinical studies and our ultimate goal of defining regimens that are more effective and hopefully in shorter durations of time. So I'm going to move us backwards along the development path, and we're again talking about the preclinical arena, where I think it's no mystery to anyone here that there is quite a bit of, fair to say, a lack of consensus about what are the best preclinical models to be working in and just what those models can offer in terms of predictive accuracy. And so being able to take a more quantitative approach and a more data-driven approach to understand those contributions and the place for those models on the critical path has been one of the major endeavors of our preclinical and clinical sciences work group. Okay, I can read it somewhat. It actually looks better than it does on my screen. So what we really have been charged to do, the mission, of course, is to try and evaluate the various models and approaches with the aim of trying to develop new ways to, new tools that can be applied to this process or new approaches to undertake in the way that we analyze and interpret those data to make these translations more seamless and more accurate. And a very early goal from our conception was to take a look at existing models and emerging models in this space to try and get a better handle on what they might contribute. And so as a part of that effort, we got together, and very much with the help of Tawanda, Deborah, Klaus, and Ann Leonards in particular, put together a landscape analysis to sort of give us a sense of how models, preclinical models, are used in the field today, and just what sort of evidence base there is to support their utility in this endeavor. And we very quickly zeroed in on the hollow fiber model as a tool that was really well poised to evaluate in terms of its predictive accuracy in this drug development process. And that's not because our analysis clearly pointed this out to be a superior tool with superior predictive accuracy. It may be, but most importantly, there was a very nice and appropriate data package with which to address these questions. And that stems largely from the quantitative nature of the types of data that come from this system. So just to orient you a bit to the hollow fiber system, if it's not well familiar to you, there are really three elements here. In this bioreactor cartridge, it's a peripheral compartment with viable bacterial cultures in that space, and those are represented as some of the little dots between the, I don't even have a pointer. No, that's not a pointer. These little dots here in between the hollow fibers. So there's this peripheral space with the cultures where one can manipulate all of the culture conditions and subject MTB to at least some of the stresses that it might encounter in vitro or in vivo, excuse me. A second element is the central compartment, the vasculature, if you will, that delivers the nutrient media as well as the drug to those bacteria in the cartridge. And the third element is the system of pumps and the dilution system that allows one to dial in very precisely the preferred concentration time profile for the drug. Now that may be done to mimic precisely human pharmacokinetics or any other schedule that one wants. The other very nice feature of the model is the sampling ports. Here, both the sampling port for peripheral and central compartments where one can do repetitive sampling over time from the same system, which is really quite powerful in terms of some of the analytical capabilities that it offers. So, I alluded to the fact that there's such nice quantitative output from this model. There's just a sampling of some of the key information that comes from the model, drug concentration information over time to validate those exposures that you're after, quantitative measures of viable bacterial counts, which traditionally has been CFU counts, but now there's exploration of other measures of viable bacterial counts, but also a wide variety of other biomarkers for TB that can be examined, RNA expression, whole genome sequencing, and the like. And finally, again, in some relatively new and certainly innovative work is incorporating some elements of the host in terms of macrophage cultures into the system as well, which allows one to look at intracellular drug penetration, toxicity of drugs at certain exposures as well. So really trying to get a little bit further towards that in vivo system. And nicely, the way that this tool has been used by Tawanda and his group principally, and what really seems to offer some of the greatest potential in terms of PK/PD translation here is the quantitative output of the PK and PD, the ability to integrate that with distributions of population pharmacokinetics, distributions of bacterial susceptibilities into various simulation scenarios like Monte Carlo simulation to really predict, forecast dose response curves, target attainment proportions or probabilities that might be expected in patients and might be used for optimal dose selection. So one can develop forecast or hypotheses that then can be tested in other in vivo scenarios, be it an animal or the clinic. Resistance as well can certainly be tracked. So to provide one example, here's work again from Tawanda's group looking at PZA. Work in the system established area under the curve over MIC as a principal driver for the bactericidal effect under these acidic conditions in these hollow fibers. A target of 209 for optimal killing, so that's an EC80 or EC90, which given the penetration of pyrazinamide into the lung represents an AUC over MIC of about 12 in the serum. And so again, putting this into simulations, again using data really just from the hollow fiber system, enabled dose finding projections or predictions that enable them to look at dose ranges and the probability of attaining this target when a standard dose of 2 grams of PZA was administered to 10,000 simulated subjects. And again, the prediction here, as you can see, is roughly 60% of patients would attain the target at that dose, and one has to go to a higher dose of 4 grams in order to better optimize that target attainment. So the effort then, given that this type of data and these types of forecasts were available for several different TB drugs, the the data package was sufficient for developing for predictive accuracy analysis. And this is just, I'm not going to read through all this, but just to give you a sense, a comprehensive literature search followed with systematic review to try and summarize all of the data as well as the outcomes of corresponding clinical trials with quality of evidence scoring to help with the weighting, and then a robust statistical analysis and mainly focused in terms of the predictive accuracy on scenarios where the hollow fiber prediction predated the subsequent clinical study in order to best optimize the bias here. So in summary, in terms of those quantitative predictions that predated results coming from the clinic, there were 14. They corresponded to 8 clinical studies and 14 clinical or quantitative outcomes among those clinical trials. And the predictive accuracy, so here we're talking about the, for example, the target that was predicted to correspond to optimal killing versus the target that was associated with sputum culture conversion in the case of PZA. Was a very high predictive accuracy, almost astonishingly high, with very minimal bias. So these data were presented both to FDA and EMA, and just prior to last year's meeting, the EMA delivered the qualification opinion. I may have to turn just to make sure I can get this all right here. But the EMA did qualify this tool for drug development use. Importantly, interactions with both regulatory groups emphasize the fact that for the time being, this should be viewed as an additional and complementary tool to existing tools. That this can be used in regulatory submissions all along the development pathway for informed design of clinical trials and interpretation of those studies as well. And they had some specific recommendations for use, including the proposed use as a way of developing preliminary proof of concept for developing appropriate combinations of drugs to move forward, for selecting the desired pharmacodynamic parameters to target and the target threshold values for those parameters associated with optimal effect. And those could be used both early in the process to try and limit the amount of dosing and experimentation that has to happen in vivo, but also later in a more iterative fashion in tandem with PK or even PD results coming from the clinic. Now, what also had come to light, both within our subgroup that put together the package, but was also highlighted by the EMA, of course, is that because this is still a very early technology and because the PK/PD studies to which these results could be compared have also been quite limited in nature, there was a fair amount of additional work that will continue to be necessary to try and iteratively develop and support this tool. There was another set of funded programs that was funded by the foundation to go ahead and expand and revisit the literature and update those analyses on a regular basis. But also, and importantly, new experimentation because the model has been largely limited to a couple of academic groups. There are very important things to demonstrate in terms of interlab reproducibility and intralab reproducibility. And so Tawanda and his team have undergone a very detailed series of studies meant to address sources of variability and to examine this interlab and intralab variability. But also one of the key points that was raised in these in the process here was the fact that much of the data had been either generated or much of the analysis had been retrospective, meaning the data had been generated either after clinical studies had been performed or during the performance of clinical studies. And so a key feature going forward is obviously to evaluate the ability of this system to truly forecast outcomes for new regimens that will be moving along, either that have just entered phase 3, will be soon entering phase 3, or perhaps at a much later date even be entering in the clinic. And so what you can see here on the bottom right are some examples. Again, additional funding has been provided to look at a wide range of such regimens to really build a solid and robust data package that can be put up against clinical outcomes on an ongoing and iterative basis. So this in vitro tool had a very successful qualification effort. Again, one of the other points that was brought up in these regulatory discussions was the fact that, at least to date, one cannot view this as a model that supplants the need to go into the in vivo situation, because there may still be aspects of in vivo infection that are not yet adequately reproduced in vitro condition. So these include some of the conditions that arise on the basis of the pathology and the interaction between the host and pathogen as they develop, of course. So that may influence both the bacterial phenotype within the lesions, but also the complexities of drug distribution into such lesions that we just heard about from Ian. So there is still certainly a rationale to be looking at in vivo models in tandem with the hollow fiber system. So we've moved on next to the murine model, where a model of relapse in the murine model has been used for several really 50 years or more now to inform evaluations of novel treatment regimens in the clinic. So this model is selected, it's important to emphasize, not because it is assumed to have high predictive value or even assumed to represent human disease better than other models, but simply because the amount of data available that corresponds to clinical outcomes was judged to be sufficiently high to warrant a look. Just to remind you how this model works, mice are infected. There may be variable incubation periods, but treatment is generally started with a relatively high burden of disease. Mice may be treated for different durations of therapy and then left for 3 to 6 months beyond the end of treatment to assess the proportion that still have culture positive organs, which would be the definition of relapse. So again, this was selected mainly because of the amount of available data and because of the role that this model has played in past and present TB regimen development. This relapse endpoint is considered at the time the closest correlate of this outcome in humans, although obviously one of our aims is to try and evaluate the accuracy of that endpoint. It's also demonstrated at least a track record in being able to forecast the treatment shortening potential of really the only 2 drugs that have been shown to do so in the clinic, rifampin and pyrazinamide. So our general aim then is to quantify the predictive accuracy of this mouse TB efficacy model and to do that by estimating the treatment shortening potential of test regimens, evaluating differences in the treatment effect size, the treatment duration necessary to prevent relapse compared to a standard regimen. We follow a similar, and this is one of the nice things about the hollow fiber model having gone first. We have a paradigm and a work plan to pursue. So I'll just briefly mention the context of use, the data inventory that's been developed, and we're just at the point as to where we're developing the statistical analysis plan. We've got a terrific group of content experts that are collaborating on this. So the context of use is supposed to be simple yet comprehensive statement of the manner in which this model is going to be used and analyzed, and just what that intended application is going to be. So it's very clear where in the stage of drug development this is being used, although I think it always bears emphasis that there is the opportunity for iterative use of this model. It's not just all prior to clinical development. And so the intended application, again, is to try and calculate treatment effect sizes on the basis of the amount of treatment that's necessary to reach a certain level of cure or proportion of cure in the mice by preventing relapse. And so in particular, I'd like to thank Ann Lennertz again for the help with putting together this data inventory. So we've gone through, and in a somewhat less than wholly systematic assessment at this point, have gone out and scoured the literature in an effort to identify relapse-based studies that have been conducted in mouse models in a way to that allows a correlation to relapse outcomes from clinical trials that have looked at similar regimens in the past. And so we've identified over a dozen treatment regimen interventions, if you will, that have been studied in these clinical trials, and just have outlined some of the regimen comparisons here. We're largely looking at studies that provided the basis for short course, the current short course regimen, some of the fluoroquinolone containing regimens and rifapentine regimens that have moved into phase 3, as well as the more novel PAMZ combination. And so as time has gone on, there's been more emphasis on this relapse endpoint, so there are more data and more studies that have been done for some of the later trials, like the fluoroquinolone trials and PAMZ, for example, as well as rifapentine. We've also collected a number of other covariates, which have clearly demonstrated that there's a fair amount of variability from experiment to experiment, and a lot of the experimental details that will challenge this analysis, in addition to some of the limited data available from earlier studies for this endpoint. So as I mentioned, we're really just in the preliminary stages of asking the level, asking about just what types of statistical analyses will these data support. And will allow the most robust analysis. And that's going to be one of the main functions of our breakout session today. So if you want to come and see how that sausage gets made, you're more than welcome. So that's the end of the update here. Just wanted to leave you with a few summary points that one of the key steps, I think, to address this translational gap is to learn what data should be coming from what models and analyzed in what manner to inform the key clinical trial decisions that are going to accelerate regimen development. And wholeheartedly endorse Dan's comments that this is best done with tools that have a sound evidence base and demonstrated predictive accuracy for the context in which they're proposed to be used. And so there are a variety of reasons that I think this is important, both facilitating development, of course, and identifying gaps where new tools or innovative approaches can be brought along, but also obviously the important aspect of increasing the efficiency of these regulatory interactions. And so, I think this process has been very successful so far with the hollow fiber system, and continues to yield important new data. And we hope to have a similar experience with the sterilizing mouse model. And just last slide is a thank you to each of the sub-teams that have worked extremely hard on these efforts.